joining, and thanks for your interest in this fascinating group of organisms. Indeed, potato bugs are isopods that don't roll up, but these sort of common folksy names usually don't mean a whole lot. But anyway, so I'm the first person in this list, and Nathan is the third person, but we would very much like to give a lot of the credit to the person in the middle, Paulita de Schmet, who is a postdoc at in Belgium, University of Ghent, who did a lot of the work that we are going to present today. And so I will start with this image, which is on the website advertising this talk. And I decided to talk just a little bit about this because it very well represents some of the messages, some of the take-home messages. And one of them is that most of the terrestrial isopod species in Maryland are non-native isopods. And you see 3 of them. This is Porcelius scaber, and Nathan can correct me if I'm wrong, because he's really good at IDing things from photographs. One of the commonest one, this is Oniscidea, which is still an introduced species, a bit more rare, but you can find it under logs or different object. And the third one, I'm not sure. It's a little blurry, so I don't know if it's a, whoops, sorry. If it's a juvenile of something or a different species, but for sure it's a non-native species.
It looks like a juvenile Oniscidea.
Yeah, that's quite possible. And then this little thing down here, since we talked about coprolites, this is actually isopod poop. It looks like this little square thing. As they feeding on wood and leaf litter and all kinds of things, this is the kind of poop that they produce. So, but I'm going to, whoops, let's just go back to slideshow. So I'm going to actually talk a little bit more, not just about Maryland, just a bit of general introduction to terrestrial isopod morphology and physiology, and then zooming in gradually from North America to Maryland, and then zooming out because we want to touch upon a more recent, very concerning movement, and that is how isopods are being collected and traded for various reasons. So before I get to isopods, though, I'm a soil ecologist, really. And so I did a lot of my work, very early work on isopods, but a lot of my work on non-native earthworms here in the US. And I'm generally interested in soil biodiversity and their role in carbon cycling, leaf litter decomposition. So I always make this, show these 2 slides, whatever I'm talking about, because I want to make people aware of the beauty, the diversity, and the complexity of the soil ecosystem. And soil is often called the most complex biomaterial on Earth. The soil fauna or the soil communities are often called the poor man's rainforest, because you can't just start digging and find all kinds of organisms at every scale. You can take out a chunk of soil like this or a tiny bit of that chunk like this, and you find the same level of diversity and complexity. And that is partially because the soil itself physically complex, which of course is also caused or formed by the soil organisms themselves. So there is a lot of different kinds of pore size and grain size and all these things. And that's partially the reason why we have organisms that span over, in terms of size, 6, 7 orders of magnitude. That's one thing, the diversity. The other thing is that this diversity is very closely related to diversity of functions. And so these organisms perform important functions for the rest of the ecosystem or ecosystem services, which means functions for us humans. And so again, because of this complexity, the different size groups are important in different functions. The small things usually are more important in forming and transforming soil organic matter. The larger ones are more what we call litter transformers. So they are feeding up of leaf litters and wood and that stuff, and that kind of thing. And the really large one, which forms, underground structures, are what we call ecosystem engineers. So the isopods that we talk about today fell into this category. We generally consider them part of macrofauna, although there are some very tiny ones, and they generally fall into this group, litter transformers. So, but of course, the soil food web itself is extremely complex. They also eat each other. There are predators, parasites, all kinds of things going on. Okay. So terrestrial isopods are crustacean. Probably most of you know that. And within that, they are a suborder, the Oniscidea suborder within the order Isopoda. Most of the isopods are aquatic and most of them are marine. There are some freshwater ones. And then among the marine ones, there are quite a few which are parasites, but like this one is living in the mouth of a fish, or they're parasites of other marine invertebrates, and others, like this large one, is living on the sea floor. And they are by some considered to be the ancestors from which the terrestrial isopods evolved, because they are the ones who were not swimming, but rather kind of walking on the sea floor. We don't really know when they evolved or when they originate terrestrial isopods, and that's because they don't fossilize very well. So the earliest ones, which are, verifiably terrestrial isopods, are enclosed in amber. And by about that time, people think that they already successfully colonized lands and diversified. We, of course, don't know how many species exist. We know roughly how many has been described, and this is the number down here. And most of them, as I mentioned, are detritivores. They eat that stuff, but they eat living stuff occasionally. And as detritivores, of course, they are nitrogen hungry, so they do anything to find nitrogen supplement. Sometimes they can be cannibalistic, and they can be also used they can feed on poop of other organisms, and they can also eat that meat occasionally. So they can be used, for instance, to clean skeletons for museum specimens. So the main morphological features, at least the ones that we use to identify them on a macro scale, is what I'm going to go through now. So they generally have, a head region, what we call a perion, that's the thorax, and the abdomen, which we call pleuron usually. The perion has 7, that's a very distinguishing feature, 7 segments, and each of those segments has a pair of running legs. So terrestrial isopods have 7 pairs of running legs. Now, what's on the pleuron, if you turn them over, these are also legs, which we call pleopods. They're very similar on the female, but the first 2 pairs on the male is modified as copulatory organs. And this is really what we are using when we want to 100%, if possible, identify a species. So we need males for that. Sometimes it's difficult because there are some species which are reproducing parthenogenetically, so they just cloning themselves, and often in those cases, we don't find males in a population, or there are very few of them. So in addition, we use the shape of the head, the various features on their exoskeleton. The number of antennal segments here can be 2, 3, many. The shape of these appendages, pleutelson, and so on. So they are actually quite diverse in terms of their, body plan. And size as well. So this is probably a few centimeters long. This is probably a very small one. As you can see, these are the isopods which live on the water land interface, usually on rocky shores. Often they call sea slaters, but I'm sure they have other names as well. Incredibly fast, they can jump into the water. So I would guess this is a soil dweller because of the short antennae, the long body, and it's probably a small organism. So often we just group them together functionally in terms of their movement. And this is coming from also Helmut Schmalfuss, who is kind of one of the greatest person, working on terrestrial isopods. As all crustaceans, they grow indefinitely, therefore they have to molt. So this is what's happening here, and they re-ingest the molted exoskeleton because they are in great need of calcium. So this is how they recycle calcium that they lose during molting. So there's many morphological, behavioral, and physiological features that we should talk about in terms of how they adapted. So this is that's the fascinating thing about them. This is one of the fascinating things. Only suborder of Crustacea completely adapted to land. So they don't have to go back to water even to reproduce. So one of them obviously relates to breathing. They have to be able to breathe. And so they have these organs, which we call white organs, or, there are many names to it, which is where gas exchange takes place. The more or better adapted to land, meaning they're more independent of humidity, the more of these things they have. The other one relates to reproduction. So all crustaceans have, or I believe, brood pouches where they carry their eggs, but they either are in water or land crabs return to the water to release the young there. But terrestrial isopods take care of it entirely on land. So they have these brood pouches, and you can see the eggs here, which is very tightly, held together. And then they have their own circulatory system to keep them moist and also to nurture them. And so the eggs hatch here in the brood pouch, and they release what we call the moncas, which are sort of a miniaturized version of the species. And the numbers can be very few depending on size or the reproductive strategy, just a few, or it could be up to 100 or even more, depending on the species. So this is probably the group of organisms that has been most extensively studied in terms of adaptation to terrestrial life. And that's because the species within this suborder represent a whole gradient in terms of how well they are adapted, how dependent they are humid, moist environment from these rocky shore isopods all the way to the ones that live in deserts. So this is Hemilepistus reimuri, a species that lives in deserts in Israel and North Africa. And so it really is amazing that despite of not being completely impermeable, not having completely impermeable exoskeleton like insects do, so they do lose water, especially in the ventral region, they are able to live in a very dry, arid environment. Okay. So the diversity hotspots for terrestrial isopods is in the Mediterranean, at least as we know. So the whole area of Greece, Italy, North Africa, the Middle East has enormous number of species. This, of course, might also reflect the intensity of studying those faunas. A lot of the taxonomists are collecting there and describing species there. So a lot of unexplored areas are still, so now we have isopod taxonomists in Brazil, and they kind of, I think the number of species they describe is doubles every year. So there's a lot of unexplored diversity we are not aware of. One thing is for sure, however, North America is not the biodiversity hotspot. So what we know, and even that is just based upon really old literature, almost 100-year-old literature, is that these are the numbers that we're sort of mention. Most of these native species, especially the inland species, are living in the southern state. And so it's reflecting kind of the diversity in Central America. As you come to further north, including Maryland, native species are only found in the coastal areas pretty much, and or in caves. And so why this is the case is an interesting question by itself. Okay, but before I get to that, but we do know that they are everywhere because people are aware of them. Most people talk about roly-polies and so on, but those isopods are very likely introduced isopods. And they all came from Europe, at least these most common one, which I would say a dozen or so that we keep encountering everywhere. And so, as I mentioned, I've been studying earthworms, which are the stories very similar for them. Most of these fauna introductions, or a large part of it, happened during early colonial times when the ships have soil as ballast material. So you can see here on this picture, not very well, but these workers are shoveling soil out of the ship into the shore because laws were put in place that prohibit releasing them into the water. And so with this soil came a lot of European members of European fauna. Some made it, some didn't make it. Other modes of introductions are people, and essentially with soil, right? So people brought in their little tree seedlings or saplings or nurseries or arboretums. Plant exchanges definitely brought in both direction species that don't belong there. So that's the mode of introduction. So how do they establish themselves, or why? So one of the things, especially with earthworms, we always bring up the Ice Age. We do know, of course, that everything during the last glaciation was, eliminated. And when the glaciers retreated, plants, flora, and fauna started to move back up. Earthworms, being very slow dispersers, never really made it beyond this last glacier line. And so we invoke this when we are explaining why do we only have, native worms down here and only non-native worms in these northern regions. It's not clear what's happening with isopods because they can move, definitely. They can move much faster, so we don't know. Another aspect of it, of course, whatever we had or might have had in terms of native soil fauna might have been eliminated during these massive deforestation events. And even though the forests have regrown, after a while, maybe the fauna couldn't return or reassemble itself. So that's the establishment. Also, the establishment part, I want to add that certainly the urban areas are great places for these introduced organisms to at least have established themselves, from which then they can spread further out. And so if you ask me what are the most successful urban macrofauna groups, I would say probably earthworms, ants, and isopods. And probably most of you have them in your backyard under stepping stones or in the mulch or in the compost. So this just shows that if I put down pitfall traps in many different places in urban areas, isopods will, at least in these cases, make up about half of that fauna. So they're really doing very well. Those species are really doing very well. So introduction, establishment, and spread. So now they spread. And so there is this one really nice study that was done a long time ago that collected all historical records that show that the most commonly known roly-poly, Armadillidium vulgare, was first spotted in greenhouses. That's often the case. And from then, it spread further west and further south. This story is a bit more complicated, and I can elaborate on this if we have time in the end. Okay, so whatever is the case, what we know is that we don't know really a lot, not just about isopods, but a lot of the members of the soil fauna. And there's just not a lot of interest to do these kinds of surveys by any, I don't know, federal agency or anything like that, which is a sad thing because there's this huge knowledge gaps. We don't know what is where and what they are doing. That is not the case in Europe. So in Europe, especially after the establishment of the EU, they poured a lot of money to have these coordinated efforts to map things, map birds, butterflies, all kinds of organisms, including soil fauna. And so it's not entirely, they're not every country participate, but a lot of countries participated or participate, and they use, standardized survey techniques, and then they can build these fantastic distribution maps. In this case, this is earthworm abundance map. You can see where there is more or less. They create these atlases so that summarizes the current knowledge of certain soil fauna groups. And based upon that knowledge, they can build these biodiversity threat maps where they can sort of assess what kind of threats are in where and how the degree of that biodiversity threat. So that brings us to Maryland. So I said that there's not a whole lot going on in the US with the exception of Maryland. So this survey is very unique, I would say, at least in terms of soil fauna members. I don't think there is anything like that that has been done in any other states. And the story starts in Belgium, with that person in the middle of that list, Paul Leiter, that's him, his advisor, and these guys are his friends they were college friends or drinking buddies, I'm not sure, but they are not biologists. And so they decided that they're going to map the terrestrial isopod fauna of Belgium. And this is the standard way to do it all over Europe. So you establish this grid of 10 by 10 kilometer squares, and then you use a method to sample and identify what's there. So make observations. So for 5 years, this is what they did. They themselves went out on their own time. And also, of course, there's a lot of interest in Europe, especially Western Europe, in soil organisms for some reason by citizens, by the public. So there were public records as well. And so as a result of that, they published this atlas for the entire country. It's a small country of Belgium. And so what you do, the project is called Spinicornis. And Spinicornis is the species name of this isopod, which has this very distinctive yellow dotting on its back. For some reason, we don't understand, they are extremely common in Canada, in the eastern part of Canada, and they're moving down. And so it looks like they are invading or, I don't know, spreading downwards to the south. So now they are in Maryland as well. So you can, if you collect this data, you can upload it to this common database, and you can see this is Belgium. They completely covered the entire country with their data. And so from that 10 by 10 kilometer, which is still very coarse scale, using models, and then knowing something about the ecological needs of the species and using additional data on climate, on land cover and things like that, you can build a much finer scale map which would map what we call habitat suitability of a species. So for instance, in this case, this roly-poly is temperature is a very important variable that determines, where you find it. And so these areas are not very suitable, probably for some reason it's colder or something like that. And then because it's a model, basically you can also play with some of these variables, certain scenarios. So what if land use changes? You know, how is that map of predicted distribution would change under elevated temperature or land use change, something like that. So Politer contacted me 3 years ago and said he wants to do the same thing in Maryland. And why Maryland? Because one reason I'm here. The second reason is because Maryland is roughly the same size as Belgium. And there are some commonalities. For instance, there is a gradient just in Belgium from mountains to coastal areas. The like I said, the size is similar. The climate is not entirely the same, but still sort of on the temperate range. And so the big difference, though, is that we hardly have any native species. So all of the species that we expected to find in Maryland were non-native species. So it would be interesting to apply those models into an entirely different situation. So they arrived, so they did the same thing. They arrived, this is his family, his wife and 2 sons, now 3, because a baby girl was just born. And so they spent here for 5 months, I believe, and they completely covered the entire state. So it was an amazing thing. They camped out in weird places, and I was with them, but mostly in the vicinity of Baltimore, but otherwise they did it on their own. And then Nathan helped out. He will talk about that a little bit here and there. So it's basically in each square, you look at different habitat types, forested, coastal, if available, something which we call more open, and then anthropogenic, which could have been people's backyards or some agriculture places or the campus or something like that. So something close to people. And so the sons help him out a lot. If you have leaf litter, one way to do it is you use a sieve and you shake it, and then you pick up certain things from here. And the kids were amazing. They were so much into it. If you don't have a sieve, then you lift up things, you look underneath logs and all kinds of places. They got everywhere. So they were really, really helpful. And so all of the species that were found, a distribution map was created based upon these data. And I'm going to show you just 3. So the red dots indicate where these species have been found, so the presence. And so as a result of this big survey, 8 species proved to be new to Maryland. And these native species, only 4 of them. This is one example of that are found only on the coastal plain. But the different species saw different distribution patterns. So this one, for instance, Oniscidea, which was on the title side, it doesn't occur, or at least hasn't been found on the coastal plain. This one, Armadillidium nasatum, is interesting because it was the commonest one, and it was not your Armadillidium vulgare that people keep in their terraria and places like that. It was this one, so it's very different from what we expected. This is also a roly-poly. It's smaller, somewhat different, but definitely extremely common. So we're gonna just highlight 2 of these findings. One deals with a native species and one with a non-native species. So I'm gonna hand it over to Nathan because he's the one who collected it with Politer. So go ahead.
So I was lucky enough to be reached out to from Dr. Politer, and he was looking for a native species, Scyphacella ericolia, which used to be widely distributed within North America from Massachusetts down to Florida. But there's no recent records of observation or collection within the 21st century. So we went out to find this species we really had no hopes that we would find it. However, we went to old collection sites along the Choptank and Nanticoke rivers where we it was kind of funny because we walked down the beach and we practically lost hope. So then we just started digging up some sand at the end of this beach and found out that we were stepping on these small burrows, which can be seen to the furthest image to the right. Small burrows that we were stepping on the whole way there that contained around one specimen each of this species that hasn't been seen in, I think it was close to 100 years in Maryland. So it's a small littoral species that we have practically no ecological data on. It's been hypothesized that they will burrow during the day and then emerge during night to forage, which could be supported as we collected them in pitfall traps along the beach. But this is just show how understudied our fauna truly is, because this common native species has possibly been pushed out due to beach tourism. However, there's just been so few observations that we really can't tell.
So, so these are sand grains, which then tells you just how small these animals are. And so we just want to emphasize, if anything deserve maybe some attention in terms of protection and conservation, this could be one. So the other species is a non-native species, Chaetophiloscia sicula. And that is a species that interesting personally to me, because 20-ish years ago, we found this species somewhere in Baltimore in an urban forest, and I had no idea what it was, never seen it. It's kind of a little thing, and we knew it's something that it's not listed in North America. So it turns out to be a new species to the fauna, which we were very excited about. And so we wrote a paper. And the one thing that, among the things that excited us, is that this is a Mediterranean species. This is how its native habitats look like. So it's kind of this dry, scrubby, mostly limestone areas. And then here it is in an urban forest. I said, wow, this is so cool, and so on and so on. So enter 20 years later, and then Paul Dieter, and also I started to collect it. And then suddenly it turns out that it's much more common now. I would argue, because now it's one of the most common species in my backyard, but I did not find them in previously. So I think this species, we're pretty convinced, is spreading very quickly. This is happening in Europe as well. We mostly found them in anthropogenic habitats and surprisingly in coastal habitats. But we find them in riparian zone along Stony Run and so many other places. Okay, so then we start zooming on and Nathan takes it over again.
So this species was quite an interesting species. I found it one day along the Pocomoke River Trail in Pocomoke, Maryland. And I went on the citizen science platform, iNaturalist, which I do a lot of identifications there. And I wanted to figure out what this species was and how far has it spread within Maryland particularly. However, when I started going through a lot of the observations, I found that it had spread practically all throughout the whole United States, all the way to an observation in California. And they are so common that they were even collected by isopod hobbyists in the pet trade, which we'll discuss further on. And they were raising these species in captivity. And this is just this map is primarily based off of the work done by Politer and then iNaturalist data. So there's a good chance that the density is a lot higher throughout the whole United States.
So what Nathan is not telling you is that we have at least a hypothesis, however wide it might be. Why are they here in these various places, right? So people have, so that brings us to the pet trade business. People have thought that this species, who keep them, is a rare endemic species from Texas.
Mm-hmm.
Rizzo something or another. So they kept them they're easy to keep. And so when Nathan put the record straight and says, no, this is an introduced species from Europe, it's not impossible that they just simply go out and dump them because it's not interesting them anymore. And that's when iNaturalist people start finding them in, close to human habitats and riparian zones. So it's very concerning. Okay, so this takes us to the pet trade. So Nathan, we go through this really quickly, okay?
Mm-hmm. So within the recently around 20 years, there has been an interest in terrestrial isopods, specifically within the reptile trade. And they were used originally as a cleanup crew because they will eat feces, they'll eat decaying matter. So they were used to clean up the enclosures. However, um, they started collecting these very colorful, ornate species, and they kept them solely as basically pet display species. Next slide. Okay, here's an example of just you can buy them on Amazon. Very popular species. Porcellio scaber is a very common cleanup crew species. And then there are some more ornate or designer species such as Cubaris rubber ducky, which has been imported from Thailand. So a lot of these species have been imported from tropical areas, which could be very devastating to their populations, primarily because in Thailand and such, a lot of species are confined to limestone outcroppings, cave environments, and most species imported are undescribed, such as this Cubaris species rubber ducky here. Of course, there's been crazy interest within the last decade. Um, Here is a rough estimate based off of online scraping that it's increased by quite a large percent.
The number of species. That's from Spain, this study.
Of course, colorful genera are preferred, mostly of Mediterranean origin with the Porcelian armadillidium, which they're usually confined to mountain ranges as well. And then some species, such as the image in this figure E, is a pseudo armadillo species, which is critically endangered and restricted to only two populations on Cuba, which were illegally transported here to the United States. So that continued collection can have horrible outcomes on these species. And of course, this is again the threat of rare and endemic species. A lot of the preferred species to keep are rare because they're designer, kind of a bragging right. And often they aren't just collected once, there's recurring collection multiple times, such as with the pseudo armadillo species. They've been collected at least 5 times just to supplement different imports into different countries. A big part of this is confusion in nomenclature, which can impact having invasive species as native species, and then the corrections there. Of course, collecting wild populations, potentially wiping them out completely. There have been a few reports in Thailand of species being completely wiped out. And then, of course, introductions to new environments, which is the main issue with the pet trade. And because a lot of the species that they keep, especially from the Mediterranean, can easily be established into the United States, as could have been the possibility with Armadillium maculatum, which was recently recorded in New York.
Okay, so just a few final. So this is a recent concern, and of course, this group has not been on the radar screen of any of these pet trade agencies or conservation organizations. So nobody paid attention to terrestrial isopods. And so we're trying, there's no rules and legislation. They are local, there might be local IUCN red list, but they are not, like made it to the global red list. So we're trying to do a few things, wrote a paper under review, actually revision. We try to establish a database which at least collects all the knowledge globally of what we have about verified species and their occurrences, and the effort to establish an terrestrial isopod specialist group within the IUCN. So in summary, I would say this is an incredibly successful group that established itself on land. And we have a mixture of native and endemic species, but we don't know really a whole lot. So really there would be a need to a systematic statewide or I guess countrywide or at least region-wide survey to have a better idea who is where. And so we can maybe better understand what they're doing or predict the outcome of these different interactions. So with that, thank you. I guess Nathan and I thank you for your attention.
All right, well, thank you. You can unshare, we'll come back together, and then let me put a spotlight on both of you here real quick. Okay. All right, and we have a couple of questions already in the chat box. You can put your questions in the chat box or raise your hand and I'll call on you and you can ask it directly to our presenters. One is, there was a question about submitting the data and information to the Maryland Biodiversity Project. Are y'all linked up with them?
I believe Politer did work with the Maryland Biodiversity Project. I'm not sure if some of the data has been submitted there. I know they're supposed to import data from iNaturalist. So I've uploaded a few observations of Scyphacella arenicola that should be imported from iNaturalist, I believe.
I just want to add that this work is he's still working on analyzing the data and verifying a bunch of things. And so this might be the reason, among other things, family and other obligations that, so it's still an ongoing project. I also want to add that this whole survey included not just isopods. So alongside, we collected also millipedes, spiders, and daddy longlegs, I believe, opiliones. So there will be more maps just like this for other invertebrate groups.
Oh, that's so exciting. Timothy asked for a taxonomic key or ID guide for the Maryland isopods. I see that Rick inputted a key there from Zoo Keys.
Yes.
Do you have any other information, or is that a good resource for folks to use?
That is a good resource. It's coming from Jeff Schultz, University of Maryland, except that we are planning to make some additions, so just an amendment, because it doesn't contain all the species that has been found, but otherwise it's a good key.
Jackie asks about their approximate size. Maybe they missed that first slide that you did.
Yeah. Nathan, you want to take that?
Their size can range quite a bit, depending on species. There's relatively large species such as Ligia, which are on the coast. They can be around, I think some of them get around 20 centimeters. They can be relatively large, down to some specimens which are around 1-2 centimeters.
Millimeters.
Millimeters, yes.
We mean millimeters.
Yes. Yes, there's some very small specimens.
Yeah,
especially,
a few millimeters, really the true soil valors. Yeah.
And Tracy, the best way to get the pictures of these critters, because they move quickly and often roll up when disturbed. So, do you have any tips for that?
Nathan could answer that maybe.
I usually use deli cups to capture the individuals to keep them so you can photograph them. Some species move very quickly, such as Philoscia, and at that point, it's just pointing and shooting and hoping that you can get a good photo of them.
If you can capture them, I try to cool them because they are, not as frisky when they are cold. So if you put them to the fridge a little bit and then you take a photo really quickly before they warm up again, that might help.
So you mentioned if we wanted to go out and collect them, you were using pit traps, the sieve method, and the leaf litter, and kind of digging in the soils, looking under logs. If we're going out, are there any other places that we should be looking if we're trying to find them?
So the very small ones, we usually extract. I have extractors, which are these funnels that has a screen, so you can collect leaf litter and some soil or debris, and then there's a light bulb over that, and the light and the warmth kind of forces them to the bottom of the funnel. This, of course, ends up killing them. So we do this when we need to know what they are. So that is the best way to collect the very small ones. Other than that, yeah, just go turn over things, look into the leaf litter or save something like that. Those are the best.
Amy wants to know, what precautions do you recommend to folks in the hobby when performing substrate changes in the terraria?
Okay, I would recommend baking the substrate, heat treating the substrate if you are going to discard it. Some individuals recommend freezing the substrate. However, some terrestrial isopods can survive freezing temperatures for a relatively long period of time as long as there's moisture. So I would always recommend heat treating the substrate and then possibly even freezing it beforehand.
All right. Mike wants to know, do they have any vocalizations?
I know that I don't believe that there's any vocalizations of North American species. However, it's extremely understudied. I know almost all armadillo species, which are of Mediterranean origin, they make a squeaking sound. And I know there's some other related Mediterranean taxa that are now being discovered to have that vocalization as
some
well.
in Greece, also they are armadillo. It's either squeaking or some of them gives like a friction when they roll in or out. That sounds like they scraping something against the blackboard or something. And why they are doing it, we have no clue. Because we don't know if they have any ways of hearing, so we don't know.
That's a good research study. Okay, Mason wants to know, have genetic data been used to study the populations and history in North America?
Answer is no, but there is a proposal. Actually, Paliter would like to continue this, so he just submitted, or we submitted a proposal that includes the molecular studies comparing and introduced populations and potential sources of origin of where they might have come from. But not in Europe, there is quite a bit, actually. In France, there is an research institute, solely devoted to terrestrial isopod research, and they do a lot of genetic studies on them.
All right. Timothy wants to know, are there any isopod species endemic to the Appalachians?
I believe there's one Mitconiscus species which is cave adapted. There's been very few studies in the Appalachians on terrestrial isopods. However, given that most of our endemic species are either cave or coastal isolated, there are probably a few native species within the Appalachians.
We just don't know.
Eric asks, have the introduced species displaced any of the native species?
Well, there are no good studies to show that. I do have a hint, so a feeling about this. And many, many years ago, I was studying a similar isopod in the West Coast, Aloniscus perconvexus. It's also a sand-burrowing isopod. And so I was going to go out and collect it, and all I got was Armadillidium vulgare, so this common roly-poly on the sandy beaches. So I don't know if it's you never know when something being replaced in these places, whether it's a result of competition or people disturbing the habitat, which is probably the case for this Scyphacella, this native species here in on the East Coast. As far as the inland and other places, the assumption really is that the native species didn't exist or we don't know. So this might be one of the reasons why they are so successful. They didn't have competitors within the same niches, but really nobody has looked at it in, you know, a scientific way.
Mason also asked, is there an ethical way to keep the isopods?
I would say keeping native species, it's a thin line because keeping certain species and then transporting them and such is a little different. Keeping native species, possibly if they're not at risk of extinction, there's just a lot of variables. So I always refrain from collecting and keeping species. Some people are fine with, collecting like Armadillium vulgare from your garden and then keeping them if you want a cleanup crew. But there's just a lot of variables there, and I never really recommend keeping any species from the wild unless you're really familiar with the group.
They make a lot, as many people know, probably people telling me they use them here in schools, and they are fun, and they are, people use them in olfactory experiments or food selection experiments. These very common ones you sort of sweep up from your backyard or something. I think it's okay. I would say it's okay. You're not causing any harm. You educating, and they are fun to look at and work with. The problem is when you are targeting rare species or endangered species. So that's kind of a little bit unethical, I would say.
Doris wants to know, does the native isopod that Nathan found on the beach, does it go in the water ever?
We really don't know. The current literature suggests that they do not. However, there is records of specimens being collected from marine benthic environments. However, those were never checked. I don't believe that the specimens even exist anymore. It's just a record in the literature.
From 110 years ago. So it's, you never know. Yeah.
All right, Mason has a question about sex ratios and with temperatures. You might want to just take a look at his question in the chat box, because I'm having a hard time talking.
I'm so
It's in the
sorry.
internet.
Or Mason, can you read it out for us?
Yeah, sorry, that was actually me. I'm just like sharing a computer with Mason right now. So
Okay.
yeah, sorry. So I do some research concerning TSD reptiles, and I look at it in conjunction with fluctuating temperatures. And I know that some isopods have a bacteria which is like acted upon them or directly like influence the species to like make them basically be kind of pseudo like TSD. So I was wondering like how the increasingly like fluctuating temperatures might look for isopod sex ratios in terms of like conservation, and if anyone's like looking at that specifically.
Okay, you want me to take this?
I have some that I could say, but basically I don't know of anyone that is, but
So, okay, so I don't think that temperature has an influence of sex ratios. However, the bacterium that you talk about is called Wolbachia. It's a feminizing bacterium. It's very common, changing, I think it's making a genetically determined male into a female, but it's very common in a lot of insect species, arthropods, so it's not just restricted to isopods. And so that definitely can change the sex ratio. Sex ratio generally varies. Pretty varies among species. So some species has, it's not 50-50. That's all I can say. And it also, well, anyway, it varies. I don't think that this is gonna be the problem for the future of the species. We do know, for instance, that a female can store a sperm up to a year. So maybe you just need a female who, and then the males can go away for a year and then she's still gonna be okay. But yeah, there were a lot of, again, in this French institute, a lot of studies on Wolbachia and how it affects the sex ratio.
I skipped Brian's question earlier on. How big is the largest terrestrial isopod recorded? And then follow up, were the ancient ones large like the water ones? And are the non-native species harmful to the ecosystem? So there's 3 questions from Brian.
Okay, Nathan.
I believe the largest terrestrial isopod is a Tylo species, which reaches around 30 centimeters.
Millimeters.
I think this one was centimeters.
Oh, but that's not a terrestrial one, is it? Is it?
It is. It's huge. It's like the palm of my hand.
It's a foot then. 30 centimeters is a
Oh,
foot.
no. It must be millimeters then. But it's like as big as my palm. Wait, let me see. I have a link.
Oh, that's 4 inches, I would say. Your
Yeah.
palm is about 4 inches.
And
Or 5.
I know in the hobby there's some specimens that is around an inch that they say are the largest. However, I'd say around palm size.
As for the early colonizers, we don't know. We really don't know. I mean, obviously the ones in amber are much smaller, but we really don't know. Some of these deep sea isopods, they are, a foot or even larger for sure. But I don't know.
And are the non-native ones harming the ecosystem?
Well, this is again a question. I get this question a lot of the time in relation with the earthworm invasion. And so the answer I say, it really depends. People, do you like them in your garden? Most people do. And so in a garden, they are good. Are they good? Are earthworms, even if not native, good in, conditioning the soil, mixing the soil, aerating the soil? And the answer is, yeah, it's good. However, in the northern forests of Minnesota and Canada and so on, where there were no native earthworms, and you had a duff layer this thick that had a very diverse soil community, and depending upon, the understory vegetation dependent upon this duff layer for their mycorrhizal associations or just simply a germination of the seeds. These earthworms invade, they eat this stuff up, they completely change the soil profile. So from that point of view, they are harmful. And so for isopods, again, I can probably answer the same way. They eat everything. They are full. Often the compost is full of them, so they probably enhance the compost. Feeding all that stuff that you put in the compost. Again, nobody has studied them from this point of view, really. As the proposal that I mentioned also has some chapters that would like to aim that question.
Eric asks, for introduced species, how do you differentiate between spread and multiple introductions?
It is difficult. So one way, obviously, is doing molecular work. So look at how different or similar the populations are. So this is where I can go back to the Armadillidium vulgare introduction. I showed you this map of introduction point in somewhere in New York, and then the spread to the east and the south, to the west, sorry, and the south. That story is more complicated because this person actually, 1990s, there were no DNA sequencing then, but there were amino acid sequencing. So what he did, he was looking at the amino acid sequencing of these Western and the Eastern population. The Armadillidium vulgare is very widely distributed latitudinally from all in Europe, from the Mediterranean all to England and further. And so he also collected specimens and looked at this latitudinal variation in amino acid sequencing. And so what he found is that the Eastern population is more similar to the Atlantic population. So England, Germany, those, France, those population in terms of amino acid sequencing. The Western population is much more similar to the Mediterranean population, which is Spain and Portugal. And so that gives that, and what we know from history gives a strong support to the idea of multiple introduction. The West goes from the Spanish and the East goes from the British, or that area, the Atlantic region. This is one. The other one is sometimes, and I know that for earthworms, if you look at their distribution in North America, often they found a disjointed maps of distribution, which stops at the Rockies. So you have introduced species here, you have introduced species there, and nothing in between. That is another support for multiple introduction. Now, multiple in terms of, there were many ships coming and all over different places, went to different harbor. It's just common sense that tells us that these happened many, many times.
All right. Do we have any other questions from folks? You can raise your hand, put it in there. Let's see. Oh, okay.
Bronwyn, Bronwyn, can I make a couple comments?
Yeah, go ahead, Dave.
Okay, someone had asked earlier about Maryland Biodiversity and Project's involvement. Nathan, before Politer visited you last year, he spent 2 days with me in Harford and Carroll County, where I took him on to some farms for some field sampling. And what he told us was that he would share his data when the Atlas work is completed. And perhaps even, I think there's a book in the works, or is it just a publication?
Publication.
A publication. Okay, once that is completed. So MBP will be getting that data. And we were talking about habitat to look for. One of the places where I find some of the really interesting isopods, Haplophthalmus danicus, is under the loose bark of rotting trees. They like to feed there. And then other points, Nathan, I'll be in touch with you probably on INAT shortly.
Okay.
All right. Thanks. Great presentation. Enjoyed it.
Another question on here is, are there any known bad features for roly-polies? Disease, property damage, crop damage?
So we tend to think of them as gentle creatures who are feeding on detritus, so they really are not doing any harm. But people also, asking me, I'm growing these lettuce in my backyard, and sometimes I see them, chewing on it. Lettuce is good food. So more recently, not in North America, but there are reports from France and Argentina, or these are the ones I know of, that populations in crop fields, certain crop fields, suddenly exploded. And if they exploded, they need something to eat. So they go for crops, especially crops that recently germinated with the tender shoots and, high nitrogen and good quality food. And unfortunately, this is especially true for organic farmings, where they don't do any kind of these kinds of pest control. So their populations could be getting out of control, and in that case, they can cause damage. There are some sort of anecdotal evidence also that certain greenhouses, because they like to be in greenhouses, the temperature is good and humid. If you grow something, occasionally their population can just explode. But generally, I think they are okay.
Another question of mating behavior. She remembers reading something about species that had an observed mating behavior, and are there other behaviors that have been observed in other species?
Talking about some isopod discuss specific species. I think it was mating, other behaviors, observed practices, more than one species. So I'm not sure what you refer to. I mean, they mate like generally like insects, there's a male and female, they copulate, and the male transfer the sperm into the female. I don't know if there's anything unique about them. I know that aquatic isopods, the male often chooses the female, a female being larger, and the male carrying the female several days, if not weeks, on his back before they actually mate. I know that is happening in aquatic isopods, but I don't know of anything unique in terrestrial ones. I don't know if Nathan, you know anything.
There are some periods of mate guarding, which is where the male will, Mason, tapping behavior. The male will sometimes basically mate guard, so it will ride the female until the female molts, and then they can copulate. That's the only behavior that I'm aware of.
Yeah.
Okay. Any other questions for Nathan or Dr. Szlavecz? Well, this has been a wonderful evening. Thank you so much for taking the time to share your knowledge, expertise, and experience with our community of the curious. We'd love to have you back, maybe talking about the earthworms and other things.
Sure.
So we'd love to invite you back for another Must Learn Thursday. And again, come and visit us at the museum. We'll talk about that. Everybody, I hope to see y'all next week when we learn about the first recorded shark death in North America, which happened in St. Mary's in the 1600s. And other than that, stay well, stay curious, and stay
Before
outside.
you go, can I just say one thing? First of all, other than thanks for listening. And one more thing in the earthworm, I'm actually heading a citizen science project. We put together little collecting kits and sending out to citizens to collect on their backyard, or more preferably nearby forests or some, varied landscapes. So either when I come and next time and talk about the earthworms, or any other time, if you would like to participate, it's probably too late this year now, but we start early summer. So, let me know.
Oh, we could definitely get a big team together to get those kits out, and folks, I know that folks will be excited to participate.
Great, thanks.
All right, thank you so much, everybody, and stay well, stay curious, stay outside. Go look for some isopods this week.
Sure, they are out there. Thanks, Nathan. Thanks, everybody.
Thank you for
Bye-bye.
having me.
Thank you. Take care,
Bye.
everyone.